Bismillahirrahmanirrahim. Today we are going to learn about the mechanism of vision. The mechanism of vision actually means that how we see various objects with the help of our eyes. To know about the actual mechanism, we are going to the first slide. In this slide, we can see the sagittal plane of an eyeball. Anything which we can see with the help of our eyes should be there in the front of the cornea. This is the cornea. This is a transparent layer, and any object which we can see should be there in the front of this cornea. The reflection of that image will fall on this layer. This is in convex shape that will contract, that will converge the object towards the pupil. So the reflection of that object will fall on that this cornea, and that will travel through the cornea into the aqueous humor. From the aqueous humor, that reflection will go through the pupil into the lens. From the lens, that will travel towards the vitreous humor. From the vitreous humor, that image or that reflection will fall on the retina. This is the retina, which is responsible for the vision, which contain photoreceptor. Actually, the retina consists of two main layers. Just like the eyeball consists of three main layers. The retina is the inner layer, the second layer is the choroid, and the third layer, or the outermost layer, is the sclera. So the retina is further divided into two layers, the neural layer and the pigment layer. For example, in this diagram we can see that this is the pigment layer and this is the neural layer. I'm going to write it: neural layer and pigment layer. So the pigment layer is there adjacent with the choroid, while the neural layer is next to the vitreous humor. For example, in this diagram we can see that the neural layer is next to the vitreous humor, are adjacent with the vitreous humor, and the pigment layer over here is the pigment layer which is adjacent with the choroid, and the neural layer is adjacent with the vitreous humor. Now in this diagram we can see that the neural layer is from this area to this area. This is neural layer, which consists of three types of cells. This is one type, this is another type, and this is another type. While the uh, pigment layer consists of only melanocytes and melanin. The melanocyte is responsible for the production of melanin. This layer is adjacent with the choroid, and the neural layer consists of the first layer. This layer is called photoreceptor layer, are the rods and cone, which contain rod and cone. The second layer is the bipolar layer, which contain bipolar cells, and the third layer is the ganglion layer, which contain ganglion cells. Or so the normally what happens during vision process, the light or the reflection of the image, the reflection of the image fall on the, I'm going to detail, the reflection of the image will fall on this cornea. From this cornea it will travel to the retina. When the reflection fall on the retina, that will goes towards what, that will actually fall on the, and that will come from this side, and that will goes through the to all the way towards the rods and cones. That will go towards rod and cone. If these two types of layer, the bipolar layer and the ganglion layer, is not responsible for the reception of light, or for the reception of the reflection of the image. Okay, the reflection of the image will fall on the rods and cones, and rods and cones are responsible uh, for the vision. So the light will go through all the way towards the rods and cones. If the reflection of the light, the reflection of the image fall on this area, if they fall on the melanin, so that will be absorbed over here. But the reflection of the image will should fall on the rods and cones. If they fall on the rods and cone, so that will activate some some pigments inside the cones and inside the rod, and those pigments will be activated, and those pigment will initiate a signal that signal will be transferred toward the bipolar cell, and from bipolar cell that will be transferred towards the ganglion cell, and that signal in the form of action potential will be transferred through the axon of the ganglion cell towards the visual cortex of the brain. So simply we can see over here that three types of cones are there. The cone having green pigment is called iodopsin. The cones having red pigment is called 4-pyropsin, and the cone having blue pigment is called cyanopsin, while the rods are having a pinkish pigment that is called rhodopsin. So normally what happens, when the light fall on the retina, it means that goes through the light goes through these two layers. The light goes through these two layers and will fall on the rods and cones. The light has nothing to do with this layer, or light has nothing to do with this layer. The light has something to do with these cells. Only these cells are responsible for the reception of light. So the light will fall on the cone, or light will fall on the rod. So when the light fall on the cone, or on the rod, for example, this is the rod, and rod and cones uh, consist of uh, inner layer and outer layer. This is the outer layer and this is the inner layer. The outer it also consists of cell body, and it also consists of synaptic end. So the rod and cone consist of large number of discs. The rod and cone consist of large number of discs, and those discs contain the pigments, and those round about one rod contain 1,000 discs, or one cone also contain 1,000 discs, and within 1 hour 3 discs are destroyed. 3 discs are destroyed, and also the formation of 3 discs also take place inside these rod. Within 1 hour 3 new discs are formed, and 3 discs are destroyed. So what normally happen, for example, the light goes through the bipolar and the ganglion layer, and that eventually reaches over here to the rods. What will happen, this light will fall on that will fall on the membrane of the rod, and through the membrane it will travel toward the disc. On the disc there is a pigment, and this pigment is called rhodopsin. This pigment is called rhodopsin. In the case of cone, that is cyanopsin or iodopsin, or 4-pyropsin. So normally this rhodopsin, I'm going to write it. The rhodopsin consists of two parts: opsin, a protein that is called opsin, and cis-retinal. So the combination of cis-retinal and opsin make what? Rhodopsin. So this rhodopsin, when the light fall on this rhodopsin, the cis-retinal of the rhodopsin is converted into trans-retinal. The cis-retinal of the rhodopsin is converted into trans-retinal, and that trans-retinal, when the cis form of the retinal is converted into trans-retinal, the opsin is separated from this retinal. Once again, when the light fall on the rhodopsin is the combination of cis-retinal and opsin. When the light fall on this rhodopsin, the cis-retinal is converted into trans-retinal. So when the cis-retinal is converted into trans-retinal, this retinal is separated from the opsin. The transretinal is separated from the opsin. This requires cis-retinal to be combined with the opsin. Only cis-retinal can combine with the opsin. When cis-retinal is converted into transretinal with the help of light, that transretinal is separated from the opsin and opsin is separated. So this cis-retinal is converted into opsin, into transretinal goes separated, and the opsin goes separated. So the rhodopsin is converted into transretinal and opsin. That opsin further initiate a process that activate an enzyme, and that enzyme is responsible for the breakdown of cGMP. This red color represents cGMP. So the opsin initiate a process, and that process activate an enzyme, and that enzyme is responsible for the breakdown of the cGMP. Now, what does the cGMP do? The cGMP it maintain the sodium channel open. The cGMP is the ligand of the sodium channel, and this always maintain the sodium channel open. The sodium channel which are there in the membrane of the rods are in the membrane of the cord of the cones. So it with each sodium channel, normally when our eyes are closed and we do not see any object, in that process the cGMP is combined with this sodium channel, and sorry, and the influx of sodium is the and there is the influx of sodium from the outside environment into the inside environment normally. But when the opsin initiate or activate an enzyme, and that enzyme break down the cGMP, then the cGMP is not able uh, to mm, to maintain this channel open. So this channel is closed. When this cGMP is break down or destroyed, the channel is open, the channel is closed. And when the channel is closed, there is no influx of sodium from outside into environment to inside environment, and eventually the inside environment of the rod or inside environment of the cone become less positive and more negative. Normally there is something weird about the rods and cones, and which is not just like normal neurons or normal nerve cell or, not, or normal muscle cells. In the muscle cell or in the neurons, the when neurons or muscle cell are at rest, that is showing -70 or -90 uh, electric potential. But but the rods and cones, when the rods and cones are at rest, they show -30 to -40 action electric potential. These show -30 to -40 electric potential, unlike the muscle cell or neuronal cells. So this in this diagram we can see that when this cGMP is break down, this channel is closed. When this channel is closed, there is no influx of sodium to the rods or to the cones. So what happens? This make the internal environment more negative. So the electric potential drop from -30 to -70. This drop from -30 to -70, and when this drop from -30 to -70, what happens? This is very weird about the rods and cones, because normally this is in the form of activation. When this rod is activated, when there is image, when the reflection of image fall on this rod of scene, and that rod of scene close this channel, and when this channel is closed, the uh, the electric potential is converted from -30 to -70. I have written 40, but this is actually 30. From -30 to -70, and when this reaches to -70, what happens? This rod, this this synaptic terminal of this rod uh, do not secrete any neurotransmitter. This do not secrete any neurotransmitter. So you should you should focus on this diagram, that normally when your eyes are closed you do not see any object. you when your eyes are closed you do not see any object. In that condition the rods and the cones have electric potential minus 30, and when it has electric potential minus 30 these rods and cone are continuously secreting neurotransmitter, and that neurotransmitter is glutamate, that is inhibitory neurotransmitter. When your eyes are closed these rods and cones will continuously secrete what these will continuously secrete inhibitory neurotransmitter towards the bipolar cell. Why? In the form of no vision, in the form of closed eyes, why these secrete the neurotransmitter? Because the bipolar cells are also very weird. The bipolar cells have a speciality that these will initiate a signal automatically by itself if these are not inhibited. If the bipolar cells are not inhibited, these bipolar cells will initiate a signal, and that signal will be transferred towards the ganglion cells. If these are not inhibited, then these bipolar cell will initiate a signal, and that signal will be transferred towards the ganglion cell. But in the case of inhibition, when these are not inhibited, when these are not inhibited, when there is an image, when there is the reflection of image, when the reflection of image fall on the rods, or when the reflection of image fall on the cone, so what happen, these will initiate a signal, and that signal will convert the electric potential from minus 30 to minus 70. And when the electric potential reaches to minus 70, there will be no secretion of the neurotransmitter, and when there is no secretion of neurotransmitter there will be no inhibition of this bipolar cell. And when this bipolar cell is not inhibited, what will happen, this bipolar cell will initiate a signal, and that signal will lead to the secretion of neurotransmitter over here, and that neurotransmitter will activate the ganglion cell, and that ganglion cell will form an action potential towards the axon, and that axon, these axons will combine together to make optical nerve, and that optical nerve will goes towards the thalamus, and from thalamus that will goes towards the visual cortex, and in the visual cortex you will see an image. So what normally happens when uh, for example, in this diagram you can see an object in the front of the eye. So the reflection of image fall on the cornea. This reflection of fall on the cornea, through the cornea it traveled into the aqueous humor. From the aqueous humor it traveled to the pupil into the lens, and from the lens it traveled to the vitreous humor, and vitreous humor, the upper side of the object will be will fall to the lower side of the retina, and the lower side of the image, or the lower side of the object will fall on the upper side of the retina. So the image will be formed in the upside down position or in inverted position, and that image will further goes towards visual cortex, and in the visual cortex this will be rectified. This inverted image will be rectified. So normally what happen, when the light, or the reflection of light fall on the rods and the cones, the rods and as I have told you that the cones are of three types, the green and the red and the blue. So if there is absence of green cones inside the retina, it means that the individual is having green color blindness. And if the individual is having red cones absent, so that individual is having red color blindness. That individual will not be able to see red object, and in the case of green color blindness, the individual is not able to see green objects. This and the absence of blue cones is not is almost negligible, but majority of the individual are having the red and green color blindness. And if there is night blindness, in the case of night blindness, the the rod of scene inside the rod cell is in minute amount inside the rod. When there is minute amount of rod of scene formation occurs inside the rod, then the individual is not able to see objects in dim light or after evening. That condition is called night blindness. So what normally happen when the light when normally the rod is not activated, when your eyes are closed, then there is no light will fall on the pigment. When no light is fall on the pigment, the sodium channel remain open, and when the sodium channel remain open, what happen, the internal environment is more positive comparatively. So the it will show the electric potential will show from -30 to -40 normally when your eyes are closed and you do not see any object. But the light fall on the uh, when the light fall on the rod of scene, or when the light fall on the iode of scene, or any pigment of the cone, this will convert retinol the cis-retinal into trans-retinal, and the opsin will be separated, and that opsin will activate an enzyme. That enzyme will break down the cGMP, and that cGMP will not able to make or to maintain this open channel. So this channel will be closed. When this channel is closed, there is no influx of sodium inside this cell. So when there is no influx of sodium, the internal environment become more negative. When the internal environment become more negative, the electric potential is shifted from -30 to -70. When the electric potential is shifted from -30 to -70, what will happen? This will uh, this will eventually uh, this will eventually lead to the no secretion of neurotransmitter. The neurotransmitter will not be secreted anymore, or negligible amount of neurotransmitter will be secreted. When the neurotransmitter secretion is stopped, what will happen? When the neurotransmitter secretion is stopped, what will happen? When the neurotransmitter secretion is stopped over here, these bipolar cell will automatically be activated, and these bipolar cell, the activation of these bipolar cell will initiate a signal that will secrete the neurotransmitter over here, and those neurotransmitter will create an action potential inside the ganglion cell, and that ganglion cell will transfer the signal towards the thalamus, and then to the visual cortex, and inside the visual cortex the image will be formed. So once again, very carefully you should listen that when there is no image, when your eyes are closed, then these bipolar cells are continuously inhibited by rods and cones. In the case of no vision, in the case of closed eyes, these the electric potential of these will be -30, and these will secrete neurotransmitter in large amount, and these will be continuously inhibited by these neurotransmitter secreted by rod and cone. And when these are inhibited, this will not lead to formation of any signal toward the ganglion cell. But when you see any object, when your eyes are open, in the case of open eyes, these will be activated, and when the signal or when the pigments are activated inside the rods and cones, what will happen? This will initiate a signal. This will convert the electric potential from -30 to -70. When the electric potential reaches to -70, what will happen? There will be no secretion of neurotransmitter, and when the neurotransmitter is not secreted, the bipolar cell will automatically be activated, and when the bipolar cells are activated, that will lead to the to the secretion of a neurotransmitter over here toward the ganglion cell, and that ganglion cell will forward the signal. So the image will be formed over there in the visual cortex. The neurotransmitter secreted over here is glutamate. That work as inhibitory neurotransmitter over here. This is something weird about the eye. And once again, the neurotransmitter secreted over here is also glutamate, but that is excitatory over here. That make action potential over here, and over here the glutamate inhibit this one. This is very weird. When the reflection goes towards the reflection of light goes toward the rod or in the cone, these pigments will be activated inside the rod and cone, and these will convert the environment to from minus 30 to minus 70, and that will lead to the inhibition of to the decreased no secretion of the neurotransmitter, and that no secretion of neurotransmitter is responsible for the vision, for the image formation. The formation of image depend upon the the level of inactivation of these cones and these rods. The level of activation or inactivation of these bipolar cells. So normally what happen, this there is another type of cells inside the neural layer, that is called horizontal cell, and this horizontal cell is connected with large number of rods and cones, and the sole function of this horizontal cell is to inhibit these cells. Why this inhibit these cells? When there is transduction, or when there is transfer of signal from the rods and cones toward the bipolar cell, then there is no further need of these cells to remain active. So these cells are responsible to inhibit these cells after the transduction of the signal. So this is just for inhibition. And there is another type of cell that is called amacrine cell, and amacrine cell basically connect the bipolar cell with the ganglion cell, and that amacrine cell is responsible for the rectification and refinement of the signal. This actually refine the signal. They share the signal between the bipolar cells and the ganglion cell. Actually, the bipolar cell is connected with the ganglion cell with the help of the synapse, but still there is need of this memacrine cell to further refine the image and to transfer the signal to the this one to the ganglion cell. Now what happens when the signal is transferred toward the ganglion, the axon of the ganglion cell combine together to make the nerve fiber, and that nerve fiber is called optical nerve. That optical nerve arises from, for example, this is optical nerve. That optical nerve will travel toward the thalamus, and from the thalamus it will go to the visual cortex. But this is not so simple. The retina is having two sides. The retina, which is adjacent to the temporal, which is near the temporal side, or lateral side, that is called temporal retina, and the retina which is near to the nose, or on the nasal side or central side, that is called nasal retina. So over here again, once again, this is nasal retina and this is temporal retina. So the axon from the temporal retina will comes out, and that will form the optical nerve, and that will travel laterally towards the geniculate nucleus of the thalamus, the geniculate nucleus of the thalamus, and from the geniculate nucleus of the thalamus, that will go to on the respective side of the um, of, of the brain toward the visual cortex. But the nasal half, or the nasal retina, from the nasal retina, the uh, the axon will goes to the opposite side of the thalamus, to the opposite thalamus, to the opposite nucleus of the thalamus. The axon from the central side or from the nasal retina will goes to the opposite side, and that will make a chiasma over here, as cross over here, and this crossover is called chiasma, and this chiasma is below the pituitary gland. Below pituitary gland, the chiasma take place, or the crossover take place. So this will lead towards the geniculate nucleus, and from the geniculate nucleus, this will go to the respective side, to the visual cortex, which is there in the occipital lobe of the cerebrum. So the image will be rectified over here, and that will, you will see the object with the help of your visual cortex. Once again, one more thing about the retina, that when the reflection of the image fall on the retina, it means that the reflection of the image goes through the to these two layers towards the rods and cones. But this normally happens inside the retina, but the case of the fovea centralis is different. For example, this is fovea. In the fovea, the reflection of the image will directly fall on the cones. That will not go through these two layers toward the cones. The reflection will fall directly on the rods, directly on the cones, because in the fovea the cones are exposed towards the cones. And I have told you that two types of cones are there in the fovea, the red and green. So the red and green cones are exposed to the image directly in the fovea. That's why the the vision of the fovea is very sharp and very acute. You can see very precisely with the help of your fovea centralis, because the reflection of the image directly fall on the cones. That will not go through the ganglion cell and then the bipolar cell, and that eventually to the rods and cones. That will not go through these two layers, because these two layers uh, are directed towards the side, are directed laterally, and uh, these cone cells are exposed directly to the image. That's why you can see very lightly, very sharply with the help of your fovea. Second thing about the fovea, about the neural layer, that the number of rods and cones are 126 million. 120 million are rods and 6 million are cones, while number of bipolar cells and number of ganglion cells are 1 million respectively. Means 1 million bipolar cell and 1 million ganglion cell, while 126 million photoreceptors. It means that one one cone is directly connected to one bipolar cell, while the bipolar cell is not connected with the one rod, that is connected with more than one rod, that is connected with numerous, with hundreds of rods. One bipolar cell is connected with large number of rods, unlike the cone, because one cone cell is connected with the only one bipolar cell, while the rods, large number of rods are connected with the one bipolar cells.